Hello children, today we are going to start a new topic, that is gametogenesis. In the first chapter you have studied about events of reproduction. Do you remember children? That is pre-fertilization event, fertilization event and post-fertilization event. Under the pre-fertilization event comes that gametogenesis and gamete transfer. So today we are going to study about the gametogenesis. What is gametogenesis? That is formation of gamete, formation of gametes. It is the formation of gametes, that gametes are haploid, from diploid germ cell. Gametes are formed from a diploid germ cells. Diploid germ cells these germ cells are diploid. What do you mean by diploid? It is having two sets of chromosome. What do you mean by two set? How the two set is formed? Now n plus n is equal to 2n. Okay, so that is haploid plus haploid fuses to form the diploid set. Okay, we are talking about human reproduction. So human beings are having 46 number of chromosome. So in the diploid set it is having 46 number of chromosome. So haploid set, that is 23 plus 23 give rise to 46 number of chromosome. So it is having one set of chromosome. This is one set of chromosome joined together to form a two set of chromosome. So we call it as a diploid set. This diploid germ cells divides and forms haploid. Haploid means it is having one set of chromosome. That means it is having 23 number of chromosome. That means it undergoes a process of cell division which is called the meiosis. It undergoes meiosis, cell division. So what is meiosis? Meiosis is a reductional division in which the number of chromosome is reduced to half. Okay, so this is how the gametogenesis is, that is gametogenesis is nothing but the formation of the gamete. Let us see what are the types of gametogenesis. We can divide the gametogenesis into two, that is spermatogenesis, and oogenesis. Spermatogenesis and oogenesis. So what is spermatogenesis? That is formation of spermatozoa. Okay, and oogenesis means formation of ovum. So formation of spermatozoa takes place in male and formation of ovum takes place in female. So in this class we will study about spermatogenesis. Okay, now first of all we will see where the spermatogenesis takes place. So we can say the site where spermatogenesis takes place. We have studied in human reproduction, that is in male reproductive system, seminiferous tubule we have already studied. So where this seminiferous tubule is located? Inside the testicular lobule, which is present inside the testis. So this is the seminiferous tubule, that is transverse section of seminiferous tubule, which is lined by germinal epithelium. Two types of cells you can see here, that is cuboidal cells and a pyramidal cell. Cuboidal cells and pyramidal cells. These cuboidal cells are known as the germ cells, or you can say spermatogenic cells. Okay, and these pyramidal cells are known as Ceratolyzer cells. Okay, so spermatogenesis takes place inside the seminiferous tubule. That means the germ cells undergo division and forms spermatid, and that is converted into spermatozoa. So this is the site where spermatogenesis takes place. Now let us study the process of spermatogenesis. formation of spermatid from germ cells. This is the first stage. Second stage is formation of spermatozoa from spermatid. So the first stage is formation of spermatid, which is haploid, from germ cells, which is diploid, and second stage is formation of spermatozoa, which is haploid, and from spermatid, which is also haploid. Under this first stage, it is again divided into 3 phases. Number 1A, we can write here, multiplication phase, B, growth phase, and C is maturation phase. Okay, again the formation of spermatid from germ cell is subdivided into 3 phases, that is multiplication phase, growth phase, and maturation phase. So we'll start with multiplication phase. Here you have seen that spermatogenesis takes place inside the seminiferous tubule, which is having germinal epithelium that develops into germ cells. This is the germ cells. Okay, which is diploid. Okay, what happened to these germ cells? These germ cells undergo mitosis division. Suppose we are taking this particular cell and it will undergo mitosis division. It will undergo mitosis division. Each and every cells undergo mitosis division. I am going to take only one cell, that is mitosis, and forms 2 cells. 2 daughter cells are produced. These 2 daughter cells are also diploid. Why? The germ cell is diploid, which is having 46 number of chromosome, and the daughter cell is also diploid, which is also, they too have 46 number of chromosome. Why? Because mitosis is an equational division, that is equal number of chromosome is distributed among the daughter cells. Again what happened? This again undergo mitosis. The cell divides into 2. It is also diploid and the cells carries 46 number of chromosomes. Okay. So this is also undergoing mitosis. These cells are known as spermatogonia. These cells are known as spermatogonia. So this is the multiplication phase. This is said to be the multiplication phase. Multiplication phase. Okay. Now what happened? Some of the spermatogonial cells keeps on undergoing mitosis division and increases the spermatogonial cell inside the seminiferous tubule. But some of the cells will remain as such. They will not undergo any mitosis division, but they increase its size, or you can say it will enter into the next phase. What is the next phase? That is the growth phase. During this process, what happened? The spermatogonial cells increases its size. The size of the spermatogonial cell become doubled. Now, it is having same number of chromosome, only the size increases, and it is diploid. The size increases due to accumulation of cytoplasm, and as well as replication of DNA takes place, but the number of the chromosome remain as such. Okay, so this phase is known as growth phase. This phase is known as growth phase. And this cell is known as primary spermatocyte. Right? So what happened in the growth phase? Only the accumulation of cytoplasm. Due to the accumulation of cytoplasm, the size of the cell increases, and as well as replication of DNA takes place. This finishes the growth phase. After that, suppose we are taking this cell. After that, this undergo next phase, that is the maturation phase. What is the maturation phase? In maturation phase, it undergo a division which is known as meiosis. Meiosis 1. As you know, meiosis is divided into two, that is meiosis 1 and meiosis 2. Meiosis 1 is said to be the reductional division. In meiosis 1 only, reductional division takes place. That means the number of chromosome is reduced to half. Suppose it is diploid, so the daughter cell will be haploid. So it produces daughter cells which is haploid. So number of chromosome will be 23. This cell is known as secondary spermatocyte. This is known as secondary spermatocyte. Soon it will undergo second meiotic division. Okay, so what happened in the second meiotic division? Again, this parental cell divides into two. Okay, and this cell also give rise to two cells. Second meiosis, we can write here as meiosis 2. This meiosis is a equational division. It is an equational division. So what happened? So as it is an equational division, the same number of chromosome reaches to the daughter cell. So it will be having haploid number of chromosome. Okay, so the daughter cells will be having 23, 23, 23, 23 number of chromosome. So that stage is known as spermatid. It is known as spermatid. Okay, now you see here we have completed the first stage. What is the first stage? Formation of spermatid. Spermatid is formed from where? From the germ cell, that is a diploid germ cells undergo division. First it will undergo mitosis division, then it after meiosis division it become a haploid cells. So what is the next stage? We have completed all the three phases. This is the multiplication phase, this is the growth phase, and this is the maturation phase. This phase is known as maturation phase. Maturation phase. So what happens in the multiplication phase? The number of the cells, that is spermatogonial cell, increases. Okay, and after that in the growth phase, some of the spermatogonial cell stops its division and accumulates cytoplasm and increases its size, and along with that replication of DNA takes place. That is known as primary spermatocyte. And after that what happened? In the maturation phase, it will undergo a reduction division, that is meiosis 1 is a reduction division in which the chromosome number is reduced to half. Okay, that means diploid cell become haploid cell. Again it will undergo meiosis 2 and forms spermatid. Okay, so what is the next phase? That is formation of spermatozoa from spermatid. So this is said to be that stage. Here no division will take place. There is no division, but there is a change in the shape of the spermatid. See this, there is a change in the shape of the spermatid. So now we can write here it as a spermatozoa. Now you see here the spermatid is circular, non-motile, but spermatozoa is oval in shape and motile. So there is a transformation took place in this region. That means a modification, we can say a metamorphosis took place. That process is known as, we can write here, spermiogenesis. Spermiogenesis. What is spermiogenesis? That is transformation of spermatid into spermatozoa. There is a transformation, there is a change, that is a metamorphosis is taking place here. The shape, function, structure, everything it is changed into a spermatozoa. Now we will see what are the changes which takes place during spermiogenesis. Spermiogenesis. So what is spermiogenesis? Transformation of spermatid into spermatozoa. During this process, what happened? So many changes we can see. So the first is nucleus condenses. Nucleus condenses, that means it is having only DNA plus basic protein. Okay. Next, this is the first change. Second change, the shape of the nucleus changes. Shape of nucleus changes. How the shape is changed? Now, the nucleus become oval in shape and the tip of the, that is anterior region of the nucleus become pointed. The anterior region become pointed and the nucleus is flat. So, the shape of the nucleus is changed. Third, Golgi body. What is Golgi body? It is a organelle which is present in the spermatid. That develops into acrosome, a structure which is present at the top of the nucleus, acrosome. Okay, now what is the fourth change? Mitochondria. So this is mitochondria. Mitochondria is also a organelle which is said to be the powerhouse of the cell. It spirally coils and forms a ribbon-shaped structure in the axial filament. Okay, so it becomes spiral and ribbon-shaped. Ribbon-shaped. It becomes spiral and ribbon-shaped. And the third and the last, that is fifth changes is that plasma membrane covers the entire spermatozoa. That is, the plasma membrane covers the entire spermatozoa. So these are the changes which takes place when spermatid is converted into spermatozoa. First change, that is the nucleus condenses. Inside the nucleus, DNA and basic proteins are there, and whatever acidic protein, RNAs are, it is removed or it is lost. And then the shape of the nucleus, which is circular earlier, that is changed into oval shape and the anterior end become pointed and it become flat. Next, Golgi body, which is an organelle present in the spermatid, is developed into acrosome. And then mitochondria, which is the power of the house of the cell, becomes spirally coiled to form a ribbon-shaped structure, and the plasma membrane covers the entire spermatozoa. Now you have seen that these are the changes in the process of formation of spermatid into spermatozoa, and the process is known as spermiogenesis. There is one more term, that is spermiation. spermiation. What is spermiation? Spermiation means, let me show you with the help of a diagram. Suppose this is the seminiferous tubule, okay, and you can see here germinal epithelium. After that you can see here spermatogonial cells. Here you can see a pyramidal shaped structure. This is the spermatogonia. This is a spermatogonia. And this is Ceratolyzer cell. Okay. So what happened during during the process of spermatogenesis? Some of the spermatogonia get embedded in the Ceratolyzer cell. What do you mean by embedment? Means it get attached with that of the Ceratolyzer cells. Okay. Why? Because it is taking the nutrition from the Ceratolyzer cells. For that process, it get embedded with the Ceratolyzer cells. So after the process of spermatogenesis, when it completely develops into spermatozoa, the head of the spermatozoa, which is embedded in the Ceratolyzer cells, move out of the Ceratolyzer cells and reaches into the lumen of the seminiferous tubule. This is the lumen of the seminiferous tubule, and that process is known as spermiation. What is spermiation? That is head of the spermatozoa removed from the Ceratolyzer cell and reaches into the lumen of the seminiferous tubule is called spermiation. Okay. So let us see what is the function of a Ceratolyzer cells. So what is the function of Ceratolyzer cell? Ceratolyzer cells provide nutrition to the developing spermatogonia. Moreover, it protects the spermatogonial cells and it gives mechanical support to the spermatogonial cell. And moreover, one more thing, it secretes a fluid. A fluid is secreted from that is Ceratolyzer cell, that acts as a medium for the spermatozoa to move out of the seminiferous tubule. Okay. Next, it also secretes a protein that is called androgen binding protein. This protein is called androgen binding protein. So as it secretes this protein, it helps to absorb androgens. Where the androgen, what is an androgen? Androgen is a sex hormone where it is produced outside the seminiferous tubule. Children, if you remember, we have studied that seminiferous tubule is the tears of seminiferous tubule appear in circular form. Here outside you can see the interstitial cells. This interstitial cells secretes androgen. Okay, that we have studied in male reproductive system. So this androgen which is secreted here in the interstitial cells is absorbed by the protein, that is an androgen binding protein, which is secreted from the Ceratolyzer cells. So what happened at the time? The concentration of androgen increases inside the seminiferous tubule. These are the functions of the Ceratolyzer cells. Okay, it provides nutrition, it gives protection and mechanical support to the spermatogonial cells, and it releases a fluid which acts as a medium for spermatozoa, and it also secretes androgen binding protein which increases the concentration of androgen inside the seminiferous tubule. So let us study the detailed structure of spermatozoa. This is the structure of spermatozoa. It is divided into head, neck, middle piece and tail. Over the head you can see a structure called acrosome. You know, this we have studied earlier. Acrosome is developed from Golgi body. Okay, and it contains an enzyme which is called hyaluronidase enzyme, which is commonly known as sperm lysine. So what is the function of this sperm lysine? It will penetrate the membrane of the egg during fertilization. Inside inside the head, you can see the nucleus. So we can label here as nucleus. Nucleus consists of DNA and basic protein. In the neck region, there is two centrioles are present. This proximal centriole, which is placed in the anterior region, proximal centriole, and distal centriole. Okay. What is the function of proximal centriole? This proximal centriole helps in the first cleavage of zygote. You know, when spermatozoa and ovum fuses and forms a zygote. Zygote undergo mitosis division. The first cleavage is with the help of proximal centriole. Okay. Now, what about distal centriole? Distal centriole, from the distal centriole, axial filament arises which passes through the middle piece and enter into the tail. So this is the axial filament, which is also known as axonema, Now in the middle piece you can see that is spirally, that is mitochondria, abundant mitochondria is present, and it is spirally coiled to form a ribbon-like structure. This arrangement of mitochondria, this is the mitochondria. This arrangement is known as Nebenkern. This type of arrangement is known as Nebenkern. Now, what is the role of mitochondria? It is said to be the powerhouse of the cell. That means it provides energy for the motility of this spermatozoa. And again you see, this is spirally coiled around the axial filament. So it gives energy to the axial filament that helps in the motion of this tail. Tail helps to move the spermatozoa. So this is the entire structure of the spermatozoa. Okay, now we will study when the process, that is spermatogenesis, takes place in male reproductive system. Spermatogenesis begins in human during the puberty. Puberty means, that means we can say 13 to 14 or 15 years of age. At that time, there is a hormone which is called GnRH. What is the full form of GnRH? You have studied in class 11. That is gonadotropin releasing hormone. Okay, this gonadotropin releasing hormone is released from hypothalamus. Where this hypothalamus is located? Hypothalamus is located in the brain. So this hypothalamus secretes gonadotropin and it penetrates, or you can say it stimulates anterior pituitary gland. It stimulates anterior pituitary gland. This anterior pituitary gland secretes two hormones, that is ICSH and FSH. What is the full form of ICSH? That is interstitial cell stimulating hormone. That is interstitial stimulating hormone, and FSH is follicle stimulating hormone. Okay, this ICH stimulate Leydig cells. Where these Leydig cells are located, or interstitial cells, it is located outside the seminiferous tubule. So when it stimulates these Leydig cells, it produces androgen, that is the hormone. It helps in the development of secondary sex organ and secondary sex characters. It helps in the development of secondary sex organ. What are the secondary sex organ that we have studied? Accessory duct, accessory gland, external genitalia, and secondary sex character in male is deepening of voice, broadening of shoulder, like that. So these are the character which is changes which takes place due to the secretion of androgen. Next, this follicle stimulating hormone stimulate the germinal epithelium. Where this germinal epithelium is present? It is present inside the seminiferous tubule. So it stimulates the formation of spermatogonia, which develops into spermatid. Okay, and it also stimulates Ceratolyzers. Ceratolyzer, which is also present in the germinal epithelium. So it produces two protein, androgen binding protein and inhibin. These are the two protein. What is the function of this androgen binding protein? Androgen binding protein helps in the process of spermiogenesis. That is, spermatid is converted into spermatozoa. So this helps in spermiogenesis, which is a part of spermatogenesis. Okay. Now, what is the function of this inhibin? Inhibin acts as a feedback mechanism. That means it will inhibit the secretion of ICSH and gonadotropin releasing hormone if the secretion is more. So this is the way by which hormone controls the spermatogenesis. Now we have completed the process of spermatogenesis. Here you have come across with so many terms, that is spermiogenesis, which is the entire process starting from the germ cells up to the spermatozoa is known as the spermatogenesis. Another thing, spermiogenesis, that is the last step, that is spermatid is converted into spermatozoa. And the third, last one is the spermiation, that is transformation, modification of spermatid into spermatozoa. Students, this is very important topic, examination point of view. You have to go through it thoroughly, try to make the flow chart. Okay, in the next class we will be studying oogenesis. Thank you.